Bismillahirrahmanirrahim. Assalamu alaikum. Shatreen Sawati is here from Biosciences with Shatreen Sawati with a new lecture, which is my second lecture from the coronavirus series. And as I have delivered a lecture on the history and the structure of the coronavirus in detail, now I'm going to deliver the lecture on the genome of coronavirus COVID-19 genome. And before starting my lecture, I have a very humble request to all of you. If you did not subscribe my channel, kindly subscribe my channel and like the videos and share the videos, share the knowledge. Share the videos means sharing the knowledge with others, so keep watching and keep sharing the knowledge with others. So I have discussed that I created all these lectures on the basis of the research articles, and I will give the link in the description box of all related research articles, so you can easily download all these articles, and these articles will be helpful to you if you are currently doing research on coronavirus, or you want to do the research in future on COVID-19. So now I'm starting my lecture on the genome of SARS coronavirus or COVID-19 in detail. So what is basically the coronavirus genome is? SARS coronavirus, first one, and this is now the novel coronavirus. All these have, the scientists revealed that 80%, the genome is 80% similar. So the genome of the COVID-19 and the SARS coronavirus, which was emerged in 2003 in China, and that COVID-19 emerged in 2019 recently in Wuhan, China. 80% similarities in their genome, and the genome almost contain 26 to 32 kilobase pair. So the genome of the coronavirus 2 or COVID-19 novel coronavirus is about 30 kilobase pair, and the genome is single stranded RNA, and this single stranded RNA is a positive sense RNA. And what is positive sense RNA? I will give you a little bit information about the positive sense RNA. And this RNA is similar to messenger RNA because it functions like the messenger RNA. It undergo the mechanism or process of translation and produce different types of the proteins which make the structure, and as well as the other non-structural proteins which play an important role in enzymatic activity and many other. So the genome size is one of the largest among RNA viruses. In addition to the SARS coronavirus, the genome of 6 species of the coronavirus have now been fully sequenced and reported in the gene bank, so you can easily find out the genome of you can say the sequence of these viruses easily from the gene bank. So before discussing in detail about the genome of the COVID-19, I will give you some information about the positive sense RNA, viral RNA, and negative sense. Positive sense viral RNA is like the messenger, similar to messenger RNA, and thus can be immediately translated by the host cell. But positive sense messenger RNA, like viral RNA, which is just like messenger RNA, undergo the mechanism of translation but by using the host cell. I will show you in my next lecture, inshallah, the complete life cycle of the coronavirus. So the second one is the negative sense viral RNA. Negative sense viral RNA has an ability to replicate into positive sense RNA by the enzyme RNA-dependent RNA polymerase. Negative sense viral RNA do not undergo the translation. It just produce positive sense RNA by replication by using the enzyme RNA-dependent RNA polymerase. So I think so the concept is now clear that what is the positive sense viral RNA and what is the negative sense viral RNA. So the coronavirus contain the positive sense viral RNA, which is just like the messenger RNA. So the COVID-19 novel coronavirus, other SARS coronavirus use the host cell for translation mechanism. Now you can see here in this figure, this is the positive and negative coronavirus genome RNA. One is positive and other is negative. So you can see here the positive sense RNA first replicate by the enzyme, and then after replication it produces the many positive RNA strands, and this positive strand has an ability to undergo the translation. Negative sense RNA do not undergo the translation, just positive sense RNA undergo the translation because it has an ability of translation and it act like the or similar like messenger RNA. Now it is clear. Now I'm moving towards the next slide, and you can see here all coronavirus contain specific genes. I will show you all these genes, and you can see here in this figure, this is the coronavirus COVID-19 genome, which is 30 kilobase pair, and this RNA, which is positive sense RNA, consists of, you can see here, 1 2 3 4 5 6 open reading frames. How many open reading frames? 6 open reading frames. And what is open reading frame? Open reading frame is that which undergo the which is responsible for translation, in simple word. And you can find out about the open reading frames and other related things in my lecture, which is about the gene. And I have discussed all these things in detail, and you can easily find out all these things there. So here I will not discuss the open reading frame in detail and many more. So genes, you can see here this open reading frame, which is ORF1A, B, this is for S open, this is responsible for the spike protein production. This is, you can say, you can see here E open reading frame, which is responsible for the production of the envelope proteins, and this is M, which is producing the membrane proteins, M for membrane proteins, and this is for nucleic acid capsid protein. So all these frames produces different types of protein, and from this to this, from S to N, you can see here, these are the structural protein of the virus. So these 1 2 3 4 open reading frames produces the structural protein, but ORF1A and 1B produces the non-structural proteins. And I will give you the concept of structural proteins and non-structural proteins. You can see here all these are the structure proteins, spike proteins, envelope proteins, and membrane proteins, and nucleic acid capsid proteins. So you can see here, you can easily read, I have given you the information about all these things. Genes for the major structural proteins in all coronavirus occur in the 5 to 3 order. You can see here, this is the 5 end and this is the 3 end, and S is started from this So these are from 5' to 3'. S started from that, ORF1, ORF2, and then S, and then so it started from occur in 5 to 3 order. Now here I have written a point, the precise strategy used by the coronavirus or genome replication is not yet known, but many features have been clarified or established. So moving towards the next slide, you can see here the genome has a 5 methylated cap. At the 5 end there is a methylated cap, and at the 3 end there is a polyadenylated tail. This is the methylated 5 end is methylated cap. As we know that in case of messenger RNA, cap and tail is present. So here in the case of the coronavirus genome, which is the positive sense RNA, it contains at the 5 end what? Methylated cap, and at the 3 end polyadenylated tail. Polyadenylated tail. Adenylated mean adenine, AAA, adenylated tail. So the genome organization for the coronavirus is 5-letter UTR. UTR is used for untranslated region. Replicase is used for the replicase enzyme, which is used by the virus for the replication of the negative sense RNA from the positive sense RNA. I will discuss in detail in my next lecture how the replicase enzyme work. So you can see spike protein are synthesized by this open-ended frame, and so on, E, M, and N. Now the concept is clear about the genome of the virus, which is positive sense and similar to the messenger RNA. And I have shown you that like messenger RNA, at the 5 end it is methylated, and at the 3 end it is polyadenylated tail, and the sequence of the structure gene is from 5 end to 3 end, like S, E, M, N, and so on. So now I'm moving towards and giving you the concept of the non-structure proteins and the structure protein. Non-structure proteins, which I have shown you, translated by the open reading frame 1A and 1B. As you can see here, this is the open reading frame 1 and open reading frame 2. So the non-structure proteins are synthesized here from this region. So the open reading frame 1A and 1B, which occupy the first 2/3 of the genome, it occupies the 2/3 of the genome, encodes the replicase polyprotein. And this replicase polyprotein is used by the positive sense RNA to replicate into the negative sense RNA. I will discuss in detail in my next lecture. Okay, so keep watching my lectures. The replicase polymerase, or which is polyprotein, self-cleave. It undergoes the self-cleavage and to form 16 non-structural proteins. To form 16 non-structural protein, NSP1 to NSP2. So 16 non-structural protein which act as an enzyme. Sometime, like you say that replicase polyprotein act as an enzyme, help in the replication of the positive sense into the negative sense RNA. Now it is clear what are the non-structural proteins which play a very important, which act as an enzyme and play other important function, but do not take part in the structure. Is it clear? Now the structure proteins. Structure proteins are spike protein, envelope proteins, membrane proteins, nucleic acid protective proteins. So the all these are the structure protein, because these four types of the open reading frames, you can see here, this operating, this, all these four operating frames help in the formation of the help in the translation, producing the structural proteins, spike protein, envelope protein. And I will discuss in detail about the S protein, because S protein play a very important role. And the scientists believe that in this novel coronavirus, the mutations occur on the spike proteins. So I will discuss in detail, inshallah, because all these lecture will help you to the synthesis of the vaccine. And also you can say that you can get a lot, that which part of the coronavirus we can target, which part of the coronavirus genome we can target for having the concept about the vaccine production. So keep watching all my lecture. I hope so the lecture will be clear upon you people. And if you have any question, kindly write in the comment or message me. I will try my best to give you the answer of all these questions. I hope so every point will be clear about the positive and negative and open reading frames and structure proteins and non-structure proteins of the genome of the RNA, and what is the difference between the positive sense RNA and the negative sense RNA. Just as I have discussed, as positive sense RNA act like the messenger RNA and undergo the translation, and the negative sense RNA has an ability to undergo and produce messenger RNA, like or you can say positive sense RNA, by the mechanism of the transcription. So negative sense RNA undergo transcription and the positive sense RNA do translation. This is basically the difference and the make the sense about the positive and negative sense. So thank you very much. Keep watching my lecture, share my videos and subscribe my channel. Allah Hafiz.